these are some discrete questions. This is question number 45. It asks, which Roman numeral represents the CNS integration for a pain reflex arc? So what we're shown here in this diagram is a pain reflex arc, and we're in particular interested in the central nervous system part of this arc. What is the CNS? Remember, this consists of the brain and the spine. And what we're looking at this diagram is this section right here, this is actually half of a spine. So when it asks for CNS integration, we're going to be looking for something within the spine. So if you look at number 1, right, we'll notice that this isn't a spine. This is actually a dorsal root ganglion. And that's when you have sensory neurons. The cell bodies kind of cluster in one area. That's what we're pointing out in 1. And that's not part of the CNS. This is part of the peripheral nervous system. So 1 is incorrect. We'll look at 2. We'll see that this neuron right here, this is within the spine. This is healthily within the spine. What it's doing here, it's getting that sensory neuron, taking that information, and then putting it into what seems to be here a motor neuron, right? It's acting as sort of a communicator here. All right, so this would be part of the CNS. So B is looking pretty good right now, but let's take a look at C and D. So 3, right here, this is part of the motor neuron. This is actually pointing at the, it looks like, well, specifically the motor neuron, and it's not really looking at within the central nervous system. This now looks like it's going to be a part of the peripheral nervous system, since it's taking that information, they've integrated within the spine and trying to release that information and communicating with number 4. And what is number 4? That's going to be the muscle. And the muscle is definitely not part of the central nervous system. So the only neuron here that's actually within the spine is number 2. That's where it looks like we're having that central nervous system, right, integration, which means B is likely the correct answer. Okay, this is question number 46. It asks, the resting potential of a neuron is primarily a result of blank. Right, remember the neuron, which I've drawn over here, has a resting potential across its membrane around negative 70 millivolts. And this is a result of, well, one of these answer choices here. A says release of neurotransmitters from the axon. Remember, what is the axon? This is the axon right here, the part that actually takes that electrical impulse and communicates it through the cell body and up to the axon terminal. So this is the axon terminal, right? But this electric potential is not kept by the release of neurotransmitters from the axon terminal. So let's say we have neurotransmitters here. It's not releasing these that would necessarily cause this neuron to stay at negative 70 millivolts. In fact, these neurotransmitters, when they reach, let's say, another neuron, they have that potential to actually disrupt the resting membrane potential, right? The resting potential of that neuron. So the resting potential is not from the release of neurotransmitters from the axon. It can actually disrupt or shift that potential that you have in the neuron. So A is not the right answer. B says the distribution of ions across the plasma cell membrane. Well, this actually sounds about right. Remember, there is the sodium-potassium pump that's working in these neurons to keep a certain potential, and that potential is the resting potential. Right, there's a sodium-potassium pump. What does it do? It takes that sodium, takes it outside the cell. Okay, it takes actually 3 outside the cell for every 2 potassiums that they bring in. And the sodium-potassium pump, it has to actually use energy to do this. It's moving these sodiums and potassiums against the concentration gradient. So what it likes to do, it likes to keep a lot of potassiums inside the cell, and it likes to take out the sodium, so the sodium is higher, the concentration of sodium is higher outside of the cell, right? So this is what causes the resting potential of a neuron, this distribution of ions across the plasma membrane. So this looks actually pretty good, but let's take a look at C and D. C says action of neurotransmitters at the postsynaptic receptor. So this is kind of similar to A. What is the postsynaptic receptor? Remember, the synapse is where the one neuron talks to another neuron. Okay, so this is the synapse right here, where we have that release of neurotransmitters, and it's going to bind onto receptors on the postsynaptic neuron here. So it's going to bind onto these transmitters. Well, again, like I said, what actually happens here is those neurotransmitters, they have this potential, or they, I guess more appropriately, they have a possibility of causing this neuron to fire as well, which would then disrupt the resting potential. So it's not necessarily that these neurons would maintain the resting potential or cause the resting potential. In fact, it can shift that potential of the neuron and disrupt the resting potential of these neurons. So C is not correct. And now let's look at D. It says active transport of calcium ions across the plasma cell membrane. Well, like I explained to you here, the resting potential of a neuron is primarily the result of these ions right here, the sodium and potassium ions. That's concentration gradient that the neuron likes to make. That is the root cause, or one of the fundamental reasons that we have resting potential of a neuron. And in fact, the calcium ions across the plasma membrane, what these calcium ions can do is actually help that neuron fire and release those neurotransmitters once the neuron is depolarized. So it's not necessarily the calcium ions across the plasma cell membrane that helps the resting potential. In fact, calcium is more involved in the release of neurotransmitters when that potential of a neuron is disrupted and that neuron starts to fire. So that makes D incorrect, and B our correct answer. Okay, this is question 47. It says contraction of the diaphragm results in blank. So let's quickly draw a model right here. And I'm going to make their torso very large to make my drawing easier. Okay, so let's say this is a human being for now. Remember the diaphragm, so let's draw our lungs. Okay, what is the diaphragm? It's this muscle that's usually just below the lungs right here or so. And this is responsible for our breathing. And what happens? So when we have contraction of the diaphragm, what happens? The diaphragm gets smaller. So let's say the diaphragm's now in red. It gets flatter. Okay, that's how I like to imagine it. It gets smaller. And what happens then? You are creating more volume within your intrathoracic area. So what is that area? This is the area, let's draw this in yellow, right here, right? It's where your lung is sitting, in between your rib cages. You're kind of increasing that volume, or the area or the space that you have in there. And what happens when you increase that space? You actually decrease the pressure. The pressure actually just goes down, remember? So when volume increases, pressure goes down, right? When we think of the gas laws from chemistry, it's working a little bit here. So when we increase that volume in a space and nothing else changes, you're decreasing that pressure. And when you decrease that pressure, what happens is this pressure actually becomes lower than the pressure of the air outside. So we have air out here. And because that pressure is lower, what happens when you have one area of high pressure and one area of low pressure? The air from the higher pressure likes to go down and fill in whatever space is here. And that space for us is the lungs, okay? So what's happening again, you contract the diaphragm. That increases the intrathoracic volume, which in turn decreases the intrathoracic pressure. It becomes lower than the air pressure outside. So what happens is the air gets shoved in, or actually likes to come in to try to fill in that space. And the only way it can do that is to come into the lungs, and that's what helps us breathe. So what are we looking for here? We're looking for a decrease in intrathoracic pressure, right? So we know it can't be B, and we're looking for inhalation, right? So, a decrease in intrathoracic pressure. We know it can't be C or D, and we're also looking for inhalation. That decrease in the intrathoracic pressure helps us breathe in. So A is the correct answer for this question. Okay, let's take a look at one more question here. It says, in an enzyme catalyzed reaction where enzyme concentration is held constant and substrate concentration is relatively low, which kinetic parameter will increase with the addition of more substrate? Note that other than substrate concentration, assume no other changes to reaction conditions. What we have here is essentially a definition quiz of Michaelis-Menten enzyme kinetics. So let's take a look at each of these terms. So Km, what does Km mean? Remember, this is the substrate concentration needed to reach 1/2 of Vmax. Now, this isn't to be confused with Km is equal to 1/2 Vmax. Km is a substrate concentration. So you should always be getting a concentration for this Km value that is needed to reach 1/2 Vmax for a particular enzyme and its substrate. So what does that mean? Even if you change the substrate concentration by increasing, by adding more substrate into some kind of solution with enzymes and substrates, that doesn't change the Km. This is a constant value for that enzyme. You're just adding more substrate, which will in turn allow you to reach maybe Km or surpass Km if you add enough substrate. So Km is a constant and it does not change with the addition of more substrate. What is Kcat? Now Kcat is essentially, conceptually speaking, Once an enzyme binds, and this is how I like to think about it. Once an enzyme binds onto its substrate, so let's say substrate is this red square, how fast is it able to turn it over to its product? So once that enzyme actually binds onto this substrate, how fast can it then change that substrate? How well can it change that substrate into its product? Let's say the product is this triangle here. So that's how I like to think about kcat. Again, this is a constant for an enzyme, which means that it will not change with the substrate concentration, which means B is incorrect. Now, what is Vmax? Now, Vmax is the maximum maximum rate that you're going to achieve with a certain concentration of enzymes. So when we actually look at this graphically, so let's say we have substrate concentration here, and we have velocity of certain reaction on that. What that graph is going to look like is you're going to have this increase, and it's going to taper off. And that tapering off point is because it has reached Vmax, right? And this is when all of the enzymes are completely saturated with the substrates that it can take. So even if you add in more substrate, there's no more enzymes that can accommodate those extra substrates, so that you've reached Vmax, the maximum velocity of these enzymes turning these substrates into, or that reaction going from substrate to the product. Okay, so that's Vmax. Now, this is a constant given an enzyme concentration, and it's not going to change because you add more substrate, right? Your Vmax doesn't change. You can get closer to Vmax, or maybe you're already at Vmax, but that value, that number, that Vmax value does not change just because you add or take away substrate. It is another constant, which means C is incorrect. And what we're left with is V, specifically V naught. But we'll just call this V, velocity, and this will change with the addition of more substrate, assuming that you're not too, the substrate concentration is relatively low, so as long as you're not all the way close to Vmax, increasing more substrate, right? If you're starting over here and you add a little bit more substrate to this point, you're going to increase that, reaction velocity. So that's what V naught stands for. It's the actual reaction velocity of this reaction that you're looking at. And as long as substrate concentration, again, is relatively low, as the problem states, you're going to be increasing. That kinetic parameter V naught is going to be increasing. It is not a constant. This is an actual value that you can change with the addition of more substrate. And that makes D the correct answer.